Oh, okay. Good morning, everyone. My name is Wei Xiong, a PI at Cyber who studies the auditory system. It's my great pleasure to have two prestigious neuroscientists in this morning session. They will show us how beautiful our sensory systems are. Our first speaker is Dr. Ardem Patapoutian. Ardem is known for identifying the molecules that sense temperature and the pressure involved in touch, pain, and regulating blood pressure. Ardem was born in Lebanon in 1967 and immigrated to the USA in 1986. He graduated from UCLA in 1990 with a bachelor degree and received his PhD from Caltech in 1996. He joined the faculty of Scripps Research in 2000, where he is currently the presidential endowed chair in neurobiology and an investigator of the Howard Hughes Medical Institute. Ardem is a member of the National Academy of Sciences and a member of American Academy of Arts and Sciences. He's a co-recipient of the 2020 Cavalier Prize in Neuroscience, the 2021 BBVA Foundation Frontiers of Knowledge Award, and the 2021 Nobel Prize in Physiology or Medicine, all with Dr. David Julius. So, Ardem, please.
Thank you. Thank you for the introduction. And I would like to also thank the organizers for inviting me today. Last time I was in China, it was almost 15 years ago, and Wei was my host at that time. So hopefully we can repeat that at some point.
Thank you. Yeah, my great pleasure.
Does this look good?
Yes, we can see it.
Okay, so today I thought I will give you an overview on what we've been doing the last 10 years or so, studying mechanosensation, mainly sense of touch through these ion channels called piezos. But for few of you who are already experts in this field, at the end I have some unpublished work in slides at the end talking about something related, but not exactly the same thing, still related to somatosensory system. But I'm mainly here to tell you that your sense of touch is quite amazing. Humans have their discriminatory touch abilities extend into the nanoscale. So you can detect a thickness of 1/500th of the diameter of a human hair. So that's quite remarkable. In addition to this discriminatory role of touch, we also rely on affective touch for our emotional health. And finally, for the related sense of proprioception, allows you, for example, to know where your fingers and hands and arms are in space without looking at them. So you can do simple tasks like standing up and walking, or more complex tasks like playing a musical instrument without looking at your hands. And this is a beautiful picture showing that. But I think one of my favorite pictures that shows the power of touch is this image from the United States, the White House. So this little boy has come to visit President Obama, and he tells him that his friends told him that his hair looks the same texture as the president's. And President Obama immediately lowers his head and says, "Why don't you touch it?" And this is captured very nicely, the boy touching the hair of President Obama. And of course, this is not just to sense the texture of the hair, but we're talking about that very affective nature, the intimate nature of touch. And so we often say seeing is believing, but in this case, also touching is believing. So the flip side of touch is, of course, pain, and acute pain is absolutely essential for our survival. But chronic pain, which is defined as pain when the stimulus that initially caused it is no longer there, is a major unmet medical need. All you have to do in the United States is follow the opioid epidemic the last few years to know that we don't have good medications for this. And so all of these senses of mechanical sensory mechanism, touch, pain, proprioception, many of them initiate at a sensory ganglia called the dorsal root ganglia. And these neurons just gather information from the periphery, and they just sit dorsal to the spinal cord and send their processes to the spinal cord and the brain. And one of the things that really distinguishes them is that they're extremely heterogeneous, as labeled here in different colors. Some are pain sensors, other low threshold mechanoreceptors responsible for touch and proprioceptors. If we look at the peripheral extension of these neurons, where they extend to, innervate, also distinguishes them as very heterogeneous. For example, these proprioceptors in red that I talked about innervate muscle spindles, while many of the fibers responsible for touch and pain, for example, innervate the skin. And there's beautiful different anatomy of different nerve endings, Meissner corpuscles, Merkel cell afferents, etc. Less studied, but these neurons also innervate some internal organs, such as the bladder, and we'll talk more about this. But at the molecular level, how do you sense a gentle breeze or a cactus pricking your finger? It was known for decades that there are these specialized ion channels that don't respond to chemicals or voltage, but actually respond to physical forces, such as mechanical force, pressure, or temperature. So over the last 25 years or so, the labs of David Julius's lab, my lab, and many others identified members of the TRP, trip ion channel in thermosensation. But just 12 years ago, the identity of mechanically activated cation channels responsible for touch, but for any other mechanical force in the mammalian system, at least, was still unknown. And so it was the work of Bertrand Coste, a postdoctoral fellow in my lab back about 10, 15 years ago. Now he's a professor in Marseille, came in to specifically identify these mammalian mechanically activated channels. And this is a bit of an old work, but I like to show it, especially if there are students and postdocs in the lab, because I think it highlights the importance of sometimes having a reductionist system to identify what you want to find, in this case, the receptors, that you can then go into more in vivo settings to look at it. And the two assumptions that Bertrand and I did were first to look at this mechanosensitivity, to come up with a very straightforward assay for mechanical stimulation and ion channel responding it. And the simple idea here was that you would actually come with a glass probe and push on a cell while electrophysiologically recording. This is voltage clamp currents. And this is what it looks like. So this is the recording voltage. You're coming with a pipette and actually stimulating the cell. And if this cell expresses a mechanically activated current, then you will open and you will record this current. So the second important assumption was that although we're very interested in the receptors accounting for touch and pain, as I mentioned, none of the receptors at that time for hearing, sensing blood pressure, none of the pressure sensors in the mammalian system were known. So we said, why don't we look at a very simple cell line that is responsive, and after we clone it from this easy to work with cell, we can go back in vivo and investigate it. So Bertrand screened dozens of cell lines and found that Neuro2A had robust mechanically activated currents. And then through a bunch of bioinformatic works, he found about 300 candidates. These are 2+ transmembrane domains that could be cation channels, and prioritized them and started knocking them out one by one in Neuro2A cells. And I also saw this slide because it shows a lot of persistence that it took from Bertrand to do it. This graph shows the screen that he did. So each of these are a single candidate gene knocking out, and this is the maximal mechanically activated current from Neuro2A cells. And in blue is just scrambled RNAi. And for each of these candidates, he spent about 2-3 days to get these kind of error bars. So literally 11 months of testing candidates getting no results. He finally, when he knocked down candidate number 72, he saw massive downregulation. This was unknown family 38 member A. he quickly renamed it PIEZO1 from the Greek piezi, meaning pressure. So we got really excited when he got these results. So the previous slide showed that if you knock PIEZO1 out from Neo2A cells, you don't see that much of a mechanically activated current. Here he cloned the full-length PIEZO1, and when he overexpress it in naive cells such as HEC293 T cells, you see massive, now we're talking nanoamp level currents just by expressing this. And the quantitation is shown on the right. PIEZO1 has a sister, about 40% identical with amino acid homology. And also, when you transfect PIEZO2 in cells, you see this very nice activation by simply pushing the cells. Another outstanding thing about piezo is like when we looked at the secondary structure, it is massive. So it's mouse PIEZO1 is, for example, about 2,500 amino acids. And most ion channels have these transmembrane domains, parts of the protein that traverses the plasma membrane. And most ion channels have 2 to 6 such domains. Piezo has 38. That's more than any other protein in your genome. On top of that, we've later figured out that it is a trimer. So it's a monstrous, almost 1 million Dalton protein, amino acid Dalton protein. Okay. Couple of slides on the structure. My lab, Kei Satome, together with Ward Lab, as well as Bailong Zhao's lab, who are here in the audience. It was my trainee. Now is running a very successful lab in Beijing. And Rod McKinnon all solved the structure of PIEZO1 using cryo-EM. This is showing the 3 protomers in different colors. Each of these ribbons is a transmembrane domain. You can see that the 3 protomers only really touch each other at the C terminus where the pore domain is. The rest, all these transmembranes are kind of extended into the membrane in a very unusual puckered shape. So it actually bends the transmembrane, the plasma membrane, apparently in the opposite direction. And many hypothesized that it is perfectly situated to be a mechanosensor because any stretch of the plasma membrane will flatten this protein and activate it. Indeed, work by Rod McKinnon using atomic force microscopy, but as well as by Long's lab, has shown that this flattening can occur. And this is a beautiful structure from Bailong's lab, and it's a morphing of the structure that I showed you already, which is thought to be closed, and another one in its flattened conformation. As you can see, this is a beautiful shift in the shape of the protein. I think this is very exciting work. What really still needs to be done is start looking at structural questions, not in detergents or liposomes, but in the native plasma membrane. This is something very exciting coming out for the future of the field. But the rest of the talk, I will focus on the physiological relevance of these ion channels. And we have used both human genetics as well as mouse genetics, both loss of function, knockouts, as well as gain of function, mutations that cause increased activity of the ion channels to identify what these molecules do. So we got very excited when we found that not PIEZO1, but PIEZO2 is massively expressed in these, our favorite dorsal root ganglion neurons that I told you is responsible for many of the somatosensory function. So this is RNA scope in situ hybridization in brown in the DRG section. You can see many of the neurons, especially the low threshold mechanoreceptors, express PIEZO2. So we've done a variety of genetic studies, loss of function to see what they do. And this is a very old slide, not very quantitative, but it's one of the first behavioral tests we've done with the mouse. So I like to show it. And it's not very sophisticated. We call it tape on the back. And here's a wild-type mouse with a big white tape on its back. It can obviously feel it and tries to get it off. And here's a deficient animal in PIEZO2. As you can see, it doesn't seem to care at all. Okay, so, but we've done some quantitation of somatosensation and just want to show you how profound the lack of behavior for somatosensory is. On the left is von Freiherr, so you touch their hind paw with different strengths of forces, And you can see after some force, you're on the y-axis, you're just looking at percent withdrawal. At the higher forces, 100% withdrawal. In the PIEZO2 deficient animals, this is complete knockout in the caudal part of the animal. You see almost no response. The next one is a cotton swab, very similar idea. Again, percent withdrawal is very, very low in the knockout. And the final one is the quantitation of the tape in the back of the video that I showed you just before. So how about proprioception? I think proprioception is perhaps one of your most important senses, but amazingly, most people don't even know about it. So it's this sense of where your limbs are due to innervation of these sensory neurons within muscle spindles and Golgi tendons. And this is how you can close your eyes and touch your nose, for example, because you have a sense of where your limbs are in space. And if you're talented and train a lot, you can do things like shown here in this slide. So is proprioception, is PIEZO2 required for proprioception? So in the mouse, show you a video of this HOXP8 PIEZO2 mouse where the caudal part of the animal is completely lacking PIEZO2. You can see extremely uncoordinated. So I'm showing you lots of behavior, but with most of these, we also do careful ex vivo electrophysiological studies. For example, this is work from Katie Wilkins and our collaborators showing that if you stretch the muscle, you can see this massive firing of wild-type proprioceptive single fiber endings. But in the knockout, 7 out of 10 is like the below, completely silent. We've seen one fiber respond a little bit. And so most of these activities, it seems to be completely dependent on PIEZO2. A fascinating turn of events, Alex Chesler and Karsten Bonneman at NIH identified humans that are actually deficient in PIEZO2. And remarkably, they have the same deficiencies that I've described to you in mice. This includes the lack of touch sensation, and as this video will show, lack of proprioception. So the way you show this in humans is you blindfold them and then ask them to walk in a straight line. So this individual who's PIEZO2 deficient, she's harnessed so she doesn't fall down and hurt herself. Again, is asked to walk in a straight line, something that most of us will do very easily, but you can see that she is having very much difficulty doing this task. How about pain? Acute pain is relatively normal in both mice and humans, but we're very interested in this phenomenon, which is part of a phenotype of chronic pain called tactile allodynia, which simply means that touch becomes painful. So you experience this after sunburn or injury, where just wearing a shirt could become painful. And this is a phenotype that folks suffering from neuropathic pain experience all the time. So we've tested this in mice. I'm not going to show you the data, but it's tactile allodynia in mice seems to be completely dependent on PIEZO2. But how about in humans? Again, these individuals who are PIEZO2 deficient really are an incredible resource to identify what's happening in the human and to see if what's happening in the mouse is in common with the humans. And so this is again work from Alex Chesler's lab. Remarkable study. We're so grateful for these individuals to volunteer for these experiments. And this is really simple experiments. On the arm, they're blindfolded, they don't see this, but we have a box A and B. And one of them, box B, gets the heat and capsaicin treatment, which causes inflammation and this tactile allodynia, where just touching it becomes painful. So with laser Doppler imaging, they make sure that the inflammation is really happening. So both in control individuals and loss of function individuals, you can see that by the red on the right side, that it is being inflamed by this treatment. And then it's simply just asking them, touching A and B with von Frey hairs and saying, do you feel the pain? So all the control individuals, of course, 100% of the time can distinguish that B is painful and A is not. And so they randomly guess A or B and get it 50%, right? So this, I think, is very relevant actually in the clinic. One of the big problems for pharmaceutical industry for pain was that many of the targets that were shown to be in mouse did not translate in humans. So here we have direct evidence that tactile allodynia is dependent on PIEZO2. And so this suggests that a PIEZO2 antagonist might become useful in the clinic. Of course, you might ask that given all of its functions in touch and proprioception, oral systemic antagonist probably is not very practical, but many neuropathic pain instances are very localized to the knees or elbows, for example, and topical antagonists, although more difficult, are indeed a possibility to go forward. Okay, so till now I've told you that various aspects of PIEZO2 requirement in touch, proprioception, pain, and allodynia in work that I didn't tell you, recently published by Rose Hill in my lab, showed that mechanical itch is completely actually dependent on PIEZO1, which is actually expressed at low levels in DRG, but we found that it's still playing an important role. But over the last few years, we've also gotten in this concept of interoception, which is mechanical sensation of internal organs. So although DRG neurons do innervate some of the internal organs, there's a whole other vagal system dependent on this. So for example, the nodose ganglia innervates the lung, the heart, the stomach, the gut, and you'll hear more from Charles Zucker after this. And they gather both chemical and mechanical information on these internal organs and translate this to the brain. And some of this is at the conscious level, some of it is not. So with respect to mechanosensation specifically, not only we don't know what the molecules involved in this, in some of the cases, the precise role of mechanotransduction in these different systems is unknown. So together with Steve Liberless, Alex Chester's lab, we've collaborated on a bunch of studies and finding important role of piezos in these systems as well. So for example, PIEZO2 from the nodose is important to sense lung stretch, and this actually regulates breathing pattern. You're not conscious of this, but there are vagal baroreceptors that innervate the aortic arch, for example, and sense how much there is increase in blood pressure or stretch. And based on this kind of information, regulate blood pressure as a feedback mechanism. And this seems to be depending on both PIEZO1 and 2. As we knock out both from the nodose ganglia, we see a very strong phenotype. Finally, Kara Marshall recently showed that the bladder stretch is sensed both in humans and in mice by PIEZO2. And interestingly, the sensor here is both in the neurons and the urothelial cells of the bladder. But I don't want to give the impression that all of this is solved. There's so many more questions in this area. For example, Rocio in my lab and Kara are both, Kara just moved on, has started her own lab at Baylor College of Medicine, are interested in this idea of gut mechanosensation, stomach or in the gut itself. So most of nutrient sensing is, of course, chemical in nature. But how much does the stomach stretch, for example? When you eat a large meal, you feel very full. How much does that contribute to how much you eat, when you eat? Diabetes, obesity is really not well known. And we feel like these molecules, PIEZO1 and 2, for example, are great tools, not just to study what they do, but again, to try to understand novel areas of physiology. And we're very excited for them to pursue these directions. So, you know, it's till now I focused on the sensory neurons. I'm not going to have time to talk to you about it, but it's not just neurons, but many cells in your bodies experience, sense, and response to mechanical force. Blood vessel endothelial cells experience shear stress, muscles get stretched, various cells, including red blood cells or white blood cells, get mechanically indented. And in many of these cases, once again, the molecules are not known and exact role of mechanosensation in these areas are not known. And again, piezos are kind of very useful to study these. And over the years, my lab and many others have found a variety of roles for PIEZO1 in various aspects of physiology and disease. Again, I won't show data, just a list here. For example, human genetics have told us that and we've confirmed this in mouse genetics, that without PIEZO1, the development of blood vessels during development don't form correctly. And interestingly, there was evidence decades ago that blood flow through the arteries, developing arteries themselves, dictate how these blood vessels develop. My Lungs lab and many others have shown that PIEZO1 is expressed in bone, and this is actually very important to regulate bone density. You might have heard that astronauts who go into outer space without this gravitational pressure on the bones, they lose bone density, and this might be one of the processes involved. Together with Metapatek, we've shown that PIEZO1 is expressed in the skin and actually plays an important role in wound healing and cell migration. Last but not least, my lab has focused a lot on the role of PIEZO1 in both red blood cells and immune cells. And for example, in one highlight, we've shown that people of African descent, 1 in 3 have this subtle gain-of-function allele, but in very large numbers that causes a variety of conditions. They have dehydrated red blood cells. We've shown that this is actually advantageous against malaria infection. But on the negative side, in macrophages, this causes elevated iron levels. So these piezos, again, are telling us not just about unexpected roles of mechanosensation, but I think in the next 5-10 years, we'll have some interesting clinical implications on their role. What I didn't tell you yet is that piezos are conserved across evolution. So we've mainly focused on the role of piezo in animals, and there have been some work on fly and C. elegans piezo, but they're also expressed in a variety of unicellular organisms, as well as in the plant kingdom. And I just wanted to highlight one slide of a recent study from postdoc in my lab, Ali Reza, who explored the role of piezo in Arabidopsis thaliana, a plant species. So plants also require mechanosensation. Just one example is that when the root of a plant goes into the soil and experiences a hard surface, it has to actively sense this and decide to go through it or go around it, for example. And it was thought that there should be mechanosensor at the root tip. And when Ali Reza looked at where piezo is expressed, it was perfectly expressed at the tips of Arabidopsis roots. And just showing you one piece of data, it seems to play an important role in detecting harder surfaces. So this is wild-type plants and mutant ones on the right-hand side, and they're exposed to a hard gel that they usually are not used to. And you can't see the roots of the wild-type on the left because they're actually going right through it. They're very thin. If you look carefully, you can see it. But the mutants cannot penetrate and seem to go over it towards you, and that's why you see all the roots very clearly. So I find this absolutely fascinating that evolution has worked on these ion channels to give rise to a protein that's required for hard surface sensing, for root tips, as well as touch sensing for you and me. Okay, so this was the kind of the overview of the work that we've done on piezo ion channels and mechanotransduction. In the next five minutes or so, I want to tell you some unpublished work we've done beyond piezos. And this is of the role of these DRG sensory neurons in various internal organs. So I've already told you that this nodose ganglia through the vagus innervates many of these internal organs. And this is a very exciting topic and area of research right now. But I also told you that some of these dorsal root ganglia neurons also innervate internal organs. And I mentioned one such study on bladder sensation. But I would say for the majority of these neurons innervating internal organs, their function is not completely understood. So in collaboration with Li Ye's lab, an assistant professor who joined Scripps recently in my own building, and we share a graduate student together, Yu Wang, we've tackled one of these tissues initially, and that is adipose tissue, which I find very interesting. So the textbook version of how fat communicates with the brain is that it secretes hormones such as leptin, that goes to the brain, and together with other information, it through the sympathetic motor system, it regulates adipose function, either, for example, to thermogenesis or adipogenesis. So this is the prevailing view, which there's, again, lots of studies, and all of this is great. But there's been anecdotal description of DRG sensory neurons innervating the fat. Never in mouse, for example, but in some species it's been shown to exist. But its exact role is not really understood. And we wanted to start investing some technology to try to do this very specifically. So the first question was, do DRG neurons really robustly innervate fat? And to do this, you used combination of techniques. One is AAV-driven fluorescent protein directly injecting into the DRG neurons that would be suspecting to innervate the fat. And then Lee's lab has come up with this recent hybrid clearing method and light sheet imaging. So we can, look at the whole DRGs from the cell body all the way into the fat cells. And I'll just show you the bottom line is that we see some beautiful DRG staining of both blood vessels and adipocytes shown here in blue and the projections in white, that these DRG neurons indeed innervate and come close opposition to fat cells, adipocytes. But I also wanted to take a step back, show you what the staining looks like, because it's very, very pretty. So these are intraganglionic injection of fluorescent protein. The dimensions is 1.2 centimeters that we can image the whole thing. It's pretty fascinating. You can see the bulbous DRGs, and north is the spinal cord projection, and the periphery is all the axons that innervate, and not just the inguinal white adipose tissue that we're interested in, but many peripheral organs. And so I showed you the specific fat innervation. This just shows you overview of where these neurons end up going. So yeah, we're very excited about being able to clear whole animals and see these kind of projections, and a lot of applications for this await. So for functional analysis, you again use the variety of some new techniques and existing techniques to look at this. And the idea is again to inject Cre-dependent, so floxed DTA, which is a toxin that would kill neurons in the DRGs. So again, the idea is we want to be able to ablate only DRG neurons that innervate this inguinal white adipose tissue, a beige tissue. And the things we want to avoid is deleting neurons in these DRGs that innervate other tissues, and perhaps more importantly, avoid sympathetic innervation of the fat tissue. And so this combinatorial approach that she's taken has worked quite well. And again, the DRGs get this floxed DTA, and then she had to use an evolved AAV9 derived Cre-expressing virus, which she then inject directly into the fat. So then the only things that will recombine are these sensory neurons that are innervating fat. We'd get the Cre from there. And she initially did this unilaterally, so all the animals are in internal control. So the left side gets only YFP, the right side only Cre. And what she found was actually quite remarkable. She saw, so she does this, waits 3-4 weeks, gets the fat tissue, and does RNA-seq. And she sees pretty nice upregulation of both thermogenesis and lipogenesis genes. For example, above shown by UCP1 expression. So these upregulations are reminiscent of what would happen if you were activating the sympathetic system, for example, by systemic cold exposure. Then she wanted to also find out if, as we were hypothesizing, this is really indeed due to regulation of sympathetic, so upstream of the sympathetic system. So she does the same experiment in the presence of this catecholinergic toxin that selectively denervates sympathetic neurons. And she sees, as you can see on the right side, that this upregulation is pretty much wiped out, suggesting that it is indeed downstream, sensory is upstream of the sympathetic. To also look at more regulation of the adipose, both morphology and physiology, she also did bilateral Curie recombination induced deletion of these sensory neurons innervating fat. And as you can see, she sees very strong enrichment of the beige adipocyte phenotype, as well as some increase in rectal temperature, indicating probably direct consequence of increased thermogenesis. So we're encouraged by these early experiments, and what these few experiments show that somatosensory DRG neurons, first of all, robustly innervate adipose tissue. And then what we have found is this a bit surprising role of sensory neuron in almost having a brake on the sympathetic system to regulate adipose function. So in a way, reminiscent of the baroreceptor system, for example, for the vagus system. So we're excited about this, and there's so many things to follow up on. How do these sensory neurons remodel in response to obesity, for example? And what are they really responding to? We have no idea what the agonists are, but one of the things we've already found out, linking back to the rest of my talk, is that many of these sensory neurons robustly express PIEZO2. And so one of our questions now is, why would you need a mechanosensor sensing fat tissue? So this is something PIEZO2 and many other candidates we're looking at right now. And of course, this approach that I told you can really be used for looking at many other interoceptive systems. What the system was really lacking was the specificity of how to get rid of sensory neurons or a specific molecule only to those that innervate one organ. And I think this method should be used to apply to other systems as well. Okay, so that's all I have for you today. This is just to thank the current lab members, alumni, collaborators, and the funding sources that were involved in the studies that I told you. But I just want to highlight a few names. Bertrand Coste, of course, for cloning piezo. Alex Chesler has been a very important collaborator for many of these studies. And the recent unpublished work, as I mentioned, it's my joint student, Yu Wang, together with Leah here at Scripps. Thank you for your attention.
Thank you, Ardem. So right now I have several questions from our streaming platforms. First one may be a very general question. What inspired you to explore the science, the sense of touch for years? Could you share your expectation of the medical application PIEZO1 and PIEZO2 in the future? Of course, you already mentioned the potential application, but can you just tell more your thought about it? Take some examples.
Yeah, I heard the second part well. Can you say the first few words? What were you asking about touch sensation? What was the first part of the question?
What inspired you to explore the sense of touch for so many years? Yeah.
It's rather interesting because I was a postdoctoral fellow with Lou Reichard, and there I was studying the role of the RG neuron, just the developmental biology of it. How do these neurons become different? I was fascinated how some neurons project to muscle, become proprioceptors, other become free nerve endings, but some sense temperature, some sense other. So this vast heterogeneity was what was fascinating to me. And then when I started my own lab, just realized that as interesting as developmental neuroscience is, what these neurons do is something that is extremely unique that other neurons don't do, which is have to sense physical stimuli such as cold, heat, and mechanical stimulation. And just a few years before I moved, David Julius had cloned the first TRPV1 heat sensor. And actually, as soon as I moved, Charles Zucker cloned NOMC, the first mechanosensor in Drosophila. So it seemed like TRP channels were playing a very important role in somatosensation. So we kind of went head in to try to find trip channels involved in somatosensation. So that was the entry point. After about 10 years of studying temperature sensation, many people thought that trip channels will also be the mechanosensors in vertebrates, but that wasn't turning out to be the case. And that's why we kind of switched to this new approach that I talked to you about to find them. With respect to clinical applications, it's kind of a two-pronged approach. Of course, they seem to be very important piezo channels, so there's probably great application for them. But the problem, there's twofold. One is that they're doing too much. So I feel like systemic antagonism or agonism could be disastrous for side effects. So that's been very difficult to tackle. And then the second one is more surprising. Finding good pharmacology against piezos have been a huge challenge. So I was involved with Novartis, a pharmaceutical company here in San Diego. And we screened 3 million compounds, their whole compound library, to find agonists and antagonists for piezos. And we found this one weak partial agonist called Yoda1 for PIEZO1 and nothing else. And so just comparatively, if we ran this on TRPM8 or TRPA1, we would find hundreds of legitimate hits. So that actually tells you something. For such a huge protein, it actually is very difficult to target pharmacology. To me, it says that, again, it goes back to its evolution of very specialized to be a mechanosensor. That's all it knows how to do. But we still have hope. We're still doing screens. I know other labs are as well, to come up with various, because neuropathic pain is something that, for example, really needs new treatments. And so, yeah, we'll see what the future holds.
Sure. Okay. The next question may be specific. Up to now, proprioception and light touch are mediated by piezos, but not for noxious mechanical touch. Is there specific mechanical sensitive ion channels for high threshold mechanosensation, or may be mediated by combinations of existing channels and the receptors on HTMR neuron?
Yeah, that's a question we think about a lot. When we knock out PIEZO1 and 2, and we have cloned some other putative mechanosensors, we're doing a triple knockout, we still haven't seen acute pain to go away. So I mentioned that both in humans and mice, acute pain remains in the absence of PIEZO2. And I'm at least convinced that none of the known sensors account for acute pain. And so we and many others have spent years and years trying to find this novel sensor. And we know it's there because in the PIEZO2 knockouts, if you go and record from the RG neurons, there's still some neurons that are mechanically activated, and they're in this high threshold mechanoreceptors. So how you find them is the question. Do you do similar approach to what we did, find it in cell lines? We've tried that, it hasn't worked. Do you go specifically into these neurons that have this electrophysiological activity, do single cell RNA-seq? We've tried that too. So yeah, I think it's a good question to ask, because people see talks like this and say, oh, all these things work all the time. Here's an example of a channel that we've been working on for 10 years and we haven't found it yet. But we won't give up, so we'll keep looking.
Okay, good. Maybe I will just ask the last question, because there are so many questions. I just propose two. You may choose the answer. The first one is how about the piezo behavior? How do you imagine in the zero gravities, such as in extra space? Or another question is, what are the difference of the piezo and the OSCA implants?
I don't have much to say about the first. I mean, I think the lack of course, piezo doesn't directly sense gravity. It's in the absence of gravity, you're not, putting force on your knees, so they're not expressing pressure. I would have loved to do some studies with, outer space, NASA or any other agency, but we haven't done that yet. That would be very interesting. OSCA in plants is definitely mechanosensor. We and others have found it. the mammalian homolog called TMEM63A, B, and C, heterologously, clearly mechanosensors, but no very convincing in vivo role have been found for them yet. And I think till that happens, I still call them putative mechanosensors. And again, some of my trainees like Shweta Murthy started her own lab. It's focusing on trying to find such roles right now.
Okay. Thank you, Ardem, because we ran out of time. And thank you again for your wonderful talk. Thank you, Ardem. Thank you.